Welcome back. So our next really big area of investigation is this idea of things moving. And materials move all the time. So in the air and in solution, we've got stuff that's constantly on the move. But with living things in biology, we're keen to see them moving into and out of cells, into and out of organisms. So, how does this happen? I've already used the word diffusion in the previous video without explaining it, but it's one of the ways that things move. Excuse me. So, around each of our cells, we have this plasma membrane. And the plasma membrane is really quite important, intricate device. You can see it's a very complex looking thing here in the diagram. We have two layers of a phospholipid, so it's a lipid with phosphorus. Lipids, fats in biology, so fats and oils are lipids. This is a phosphate-based lipid. And we have heads and tails on them. So you see all these beads at the top here. There's a layer of beads on the bottom. That's, each of those is the head of one of these lipids. And they have little tails dangling down. So this outside area here is water-loving. So it's hydrophilic, sorry. And so underneath here is also hydrophilic. But both the tails are hydrophobic. They hate water. Now, if you think about two north ends of magnets, if you had two north ends coming together, they push away from each other. You've done it when you're back in year 7 or whatever. This is also happening here. So this is a hydrophilic tail. That's a hydrophilic tail. They're the same. They actually want to repel each other. So this stuff here, these little yellow objects here, are called cholesterol. And cholesterols are used to help sort of lock the two layers together. So the two layers are sitting on each other and they're in a fluid motion. So it's known as actually the fluid mosaic model of the membrane. And they're moving over each other, which is great because it means the membrane can move, the cell can move, the organism has the ability to be flexible. So it's really useful. But the cholesterol's there to try and make sure that the two things don't come apart completely and the whole thing falls apart. You've also got protein channels, and there's other protein channels here. I won't go too much detail. Some of the things up here are things like self-markers. They explain that the cell belongs here. And others are places where chemicals can come and dock and pass on their information. So a whole lot goes on. The membranes are really complex and interesting organelle. But essentially all it's doing is communicating the external environment with the internal environment, stopping things coming in that shouldn't come in, stopping things going out that shouldn't go out. So, it's like our epithelial skin. It's a really control mechanism. But we do want things to come in. We do need things to come in. We do need things to go out. So how do they do this? Well, the easiest way, and the one that all organisms love because it's energy free, is diffusion. And diffusion is simply the movement of a material from an area of really high concentration. So this diagram here shows that. Lots of green stuff in solution. And it's in high concentration here. And of course over here there's a really low concentration. So that sort of gradient is quite a fast gradient. So that would move quite quickly from one side to the other until it's basically even on both sides. And things do that in water all the time. Do it in the air all the time. If I go off in the corner of the classroom, let off some perfume, you'd all eventually smell it because it'd diffuse out for the whole room. If we put a membrane in between, like this diagram here actually has a membrane in between, then it's got to cross that membrane. But lots of stuff can diffuse across. So oxygen and carbon dioxide quite happily diffuse across membranes of cells. Water moves by a process called osmosis, which is a form of diffusion. So in the case of osmosis, excuse me, we have a high concentration of dissolved materials and a low concentration of dissolved materials. And water moves from the low concentration to the high concentration of dissolved material. It's a variation that achieves the same outcome. But the variation is quite important. And one of those things I love picking up on exams. Just putting it there for now. We'll play with this idea. We'll actually go to experiments of diffusion osmosis and try and come to grips with it as, next week probably. facilitated diffusion. So some things can't cross the membrane. Some things are not lipid soluble. So this is, remember, biphospholipid layer. It's a fat layer. Some things dissolve in fat, some things don't. Glucose is not lipid soluble, but we need lots of glucose. So the concentration gradient, lots of glucose out here, not much glucose in here, exists, but it can't cross straight through. So it uses a channel to come through. Or in some cases, the substance may, in fact, have to attach to a protein which acts as a carrier through the channel. But again, they're in high concentration on the outside, low concentration on the inside. They said help getting across that membrane. It's interesting because water isn't lipid soluble. And there's quite a lot of argument these days about whether or not water actually diffuses, so osmosis, across the cell membrane or works through a protein channel. And they have found protein channels dedicated to water movement. It's not in the textbooks, lots of argument about it, but we may one day see facilitated osmosis as a form of movement, because there's a protein channel for the movement of water along a concentration gradient. Anyway, that's for the future, but it's an interesting idea, because water, of course, is not lipid soluble, we know that. The other main form is active transport. So diffusion, osmosis, facilitated diffusion, it's all free, energy free. The money, the currency in a cell's case is energy. How much energy do you need to spend to do this? And of course, you want to be as efficient as possible. So that they can conserve energy, they do. So sometimes you need to actually bring things in, either because they're so big they can't cross the membrane, or because the concentration gradient is actually working against bringing them in. So there's more on the inside of the cell than there's on the outside of the cell, but you still want more in anyway because you need it. So we have protein channels again for that purpose, and carrier proteins for that purpose, not protein channels, so you don't use protein channels. But you need to spend energy to get the stuff in. So this is the big difference. Large proteins and things need to be brought through this process because they're so big. And then, of course, there's also some interesting little adaptations of movement, often called bulk transport. It's not active transport in the sense of bringing things through with carrier proteins or through the channels. It's actually manipulating the membrane itself. So in this case here, this endocytosis is bringing things into the cell. And we have endocytosis and PINOCYTOSIS, and PINOCYTOSIS is a form of endocytosis, but it's related to liquids. So if this is a solid or liquid, the membrane encapsulates it. It's called invagination. It wraps itself around there and gathers a whole lot of it together, and that bit of membrane then nips itself off and creates a vesicle full of the stuff you wanted that's outside the cell, and you brought it in. Easy peasy. And of course, as you can imagine, the opposite is just as possible. So EXOCYTOSIS is the same process. Here's a vesicle full of, it could be a protein the cell's made. And the vesicle moves out to the membrane and releases its content into the extracellular fluid to wander off into the bloodstream and go and find some place to do whatever mischief it's meant to do. What's a good example? Could that be insulin? Yeah, let's call it insulin. So this is a beta cell in the islets of Langerhans in the pancreas, and it's releasing insulin that's been made in that cell, and that insulin's going to go out and tell the bloodstream to take up more glucose and store it as glycogen. That's its job. This is how we take, now I'm jumping well ahead. Our DNA codes for making proteins. The proteins are made by the cell, but then that protein has to be moved out. That's normally moved out by exocytosis to go off and do something else. I'm getting ahead of myself. We'll come back to that sort of stuff later. And that's the five. No, I've got one more, sorry. I'll be quiet, Gary. Haven't finished yet. I'll see you next one.